the Felix Factor with me, Felix Heron. So today we're going to be talking about digestive systems.
Digestion. Digestion.
Specifically that of humans and rats, which leads to the four main parts of digestion. First, ingestion. Putting food into your mouth, in the case of humans, or rats, or most mammals that I can think of. Secondly, digestion. Breaking down the macromolecules which you eat into smaller molecules which can then pass through your plasma membranes into your cells. 3. Absorption, which is when these smaller molecules then pass into your cells to be made into energy and ATP by your mitochondria. And 4th, elimination, which is taking the non-absorbed material out. Now you might ask, Felix, how is it that animals don't digest themselves when digesting their food? Because after all, animals and their food are both made up of the same basic substances, the same basic 20 amino acids, fats, proteins, carbohydrates, etc. Well, the answer is, partially at least, that an evolutionary adaptation of animals is that they process their food in sorts of compartments that specialize in digesting the things that are inside. For example, in the intestine, the columnar cells that line the lumen, they secrete a mucus that makes sure that there's like a little spot between. Now, during this whole digestive process, there has to be some type of food that we are digesting. Well, here's your answer. There are four main types of substance which animals digest, which they take in. The first is protein, which animals digest using protein digestive enzymes, and those become amino acids. Now you may notice that there is usually a macromolecule, in this case protein. There is a digestor, which is the digestive enzymes, and that becomes the component, which in this case is the amino acid. Now secondly, we have the polysaccharides and disaccharides, which, using the carbohydrate digestive enzymes, those turn into monosaccharides, mono meaning one, poly meaning many, and di meaning two. But the goal is to go from many to one. Next is the nucleic acid, which, as you might know, makes up your DNA, which is, of course, in plants and animals. That's what tells them what to make and stuff. Through the nucleic acid digestive enzymes, those turn into nucleotides, which are the building blocks of the DNA. And lastly, we have the fat, which, using fat digestive enzymes, what do you know, turns into glycerol and fatty acids, which are the things that make up the fats, and those can then be stored. Well, here's something to think about. Your food is never really in you until your cells absorb it. So, if you were to put some undigestible food into your mouth, you go into your mouth, down through your esophagus, into your intestinal tract, then out through your anus. But the whole time, it wouldn't really be in you, because it wouldn't really be in your cells. It's kind of a hard concept to grasp, but it's rather interesting that you're never really taking your food inside of your body. It's in the alimentary canal, which is the tube that goes through your body, one of the parts of the intestine, and that's where a lot of the absorption takes place. So let's just track the movement of like an apple or something. For this case, it's going to be a little blue circle. But so I put it into my mouth. That's the ingestion. Then my tongue pushes it back into my alimentary canal, into my esophagus, and so this then it really enters my alimentary canal there, and the way it moves down my esophagus, partially by gravity, also by peristalsis, which is a pretty cool thing. It's the smooth muscle in your esophagus goes, and it goes and it pushes the food down into my stomach. Now in my stomach there, it starts to get digested with the different enzymes in your stomach. Then, but before that, I skipped a step. There are the sphincters, which are little little doors that say, okay, we're letting food in now. Food comes in, no longer. And then on the other side of the stomach, okay, time to be digested. No more digestion. So it's kind of like letting food in and out. And it's a cool thing because it, regulates your tract. And it passes through your intestines, your, duodenum, and, things like that. And through your vertical column and stuff, descending, excuse me, and then to your anus to be excreted. So obviously a very important part of digestion is the enzymes that really do the breaking down of molecules, and especially for starches in the mouth, the salivary glands secrete these enzymes that start the digestive process in the mouth. Then there's 3 other important glands in the body. There's the liver, which makes bile, the pancreas, and the gallbladder. And these all help making enzymes and stuff to help with the digestive process. Needless to say, all of these end up in the small intestine, where they help to break down the food so it can be absorbed in the villi of the small intestine, which has little bumps which make the surface area as big as a high school basketball court. Unbelievable. Word on the internet, it must be true. But kind of interesting analogy I made is that, in the roots, you got like the big root stalk, and you got the little root projections, which are kind of the villi, the root hairs, which are like microvilli. Yeah, maybe. But what I do know is that there are 4 types of basic molecules, things that get digested in your small intestine. First, you got the carbohydrates. These either start out as polysaccharides or disaccharides. But the goal is, of course, to get them to monosaccharides. So if it starts out as a polysaccharide, you got the amylase, which comes from the pancreas, and that makes it into maltose. And then from the maltose, you got the maltase, and sucrase and the lactase, and all those good digestive enzymes. These make them into monosaccharides. And as you'll notice, if you have something like maltose, the thing that digests maltose is a maltase. Maltose is sugar, and maltase is enzyme. It's a good way to remember that kind of stuff. Next, we have the proteins. And the proteins, which are, of course, polypeptides. These get digested by things called trypsin and chymotrypsin. And these become smaller polypeptides. Then they are digested by aminopeptidase, carboxypeptidase, and dipeptidase to become amino acids. And what you'll notice is, of course, the A's and the peptides, peptidase, peptides. I don't even know what I'm saying anymore. But yeah, then we have the nucleic acids. This is, of course, the things that make up the DNA. These are digested by nucleases, which become then nucleotides, and then they just get digested eventually into the bases, sugars, and phosphates, which we know make up your DNA and RNA. So it's good. And then lastly, we have the fats, and using bile salts, these made into fat droplets, which are, of course, emulsified. Using lipase, these then become fatty acids and glycerol. One problem with fat is that it's not soluble, so it's a little bit harder to digest, but gotta figure it out pretty well. Because it is not, of course, soluble, it's hard for it to be digested until it hits the small intestine. And emulsification is really just the breaking up of the larger fat droplets into smaller ones. So now that we understand backwards and forwards, it's time to compare the different types of digestive systems in different animals. I think this calls for a celerity round. Celerity round! So, first, herbivores. Now, herbivores, as you know, eat lots of plants, which means they eat lots of cellulose. And cellulose is very hard to digest. So, they, therefore, have very long small intestines. For example, this bird. Small intestine, you can see, it's pretty long, because it has to digest all the cellulose it eats and all the seeds it eats. Therefore, herbivores have longer digestive tracts. It takes longer for them to digest stuff. Then, we have a meat eater, like this fish. You can see, not that long of a digestive tract, mostly because its small intestine can do its work pretty fast, because it doesn't take as long to digest fast, because they don't have secondary cell walls and stuff, which takes a long time to digest. Then we have animals like the ruminants, which are like the cows and sheep and deer and stuff. 4 chambers of stomachs, which basically means they can chew their cud. And you might have heard this before, which is when food passes from one stomach to another, and they chew it again, just to basically get all that stuff digestible for the animal. This is helpful because, especially in the herbivore animals, there are these things called a cecum, which basically houses lots of little bacteria, which help to digest the the material that is brought in, and occasionally these things are digested. But as you can see, like in koalas, they have very long cecums, which means that they can get a lot of digestion done very easily. And it's kind of gross, but some animals like rabbits, they eat their food and poop it out, and they eat it again so they can actually get every single nutrient digested. I would rather just eat more food. That's kind of gross. So I have a pretty authoritative understanding about all this digestion stuff. It's time to talk about the cavities from a different perspective. This being an iBook made by Senor Hermanson. So the first thing that we're going to do is to compare the different body cavities between a rat and a human. So starting at the top, we have the nasal cavity, which is of course the nose and all the things therein. The two main things we find we have in our nasal cavity are our sinuses, which are basically like this part here, which help our voice resonate among other things, which are currently being debated by scientists. Also, we have our olfactory receptors. So these olfactory receptors are in your nose, and they're receiving something. What could it be? Smell. Yes. It detects odor. There you go. That's what's in your nasal cavity. And funny enough, these same two things exist in rats and humans alike. Now moving down if you're a human, and moving sideways if you're a rat, we have the oral cavity, which is right here. And in this amazing cavity, we have the teeth, which are used for grinding up food mechanically. We have the tongue, which is used to push that food into the esophageal tract. Next, we have the salivary glands, which secrete digestive enzymes into the mouth to help digest things like starches and carbohydrates. And, of course, we have the hard and soft palates and the taste buds, which are used for tasting things. And these are the same, exact same, with, of course, minor differences, perhaps in location, but the same glands and the same tissues and organs exist in rats and humans. Amazing. So the next cavity is the thoracic cavity. And you might think, well, I've heard of thorax before. What is that? Well, that is a part of the animal that is this portion here. And what do you know? The thoracic cavity is exactly that. That's the cavity that contains the organs there, which is, of course, the heart, which pumps our blood, the esophagus, which transports our food, and the lungs, which help us take in oxygen and put out carbon dioxide, much of other gases, in order to fuel cellular respiration, which is beautiful. And the same exact thing in humans and rats. Unbelievable. So the next cavity is the abdominal cavity. And as you know, the abdomen is right here. It's going to contain all our main digestive organs. Probably. Oh, seems as if it does. So these organs contained within are the liver and gallbladder, along with the pancreas, which are the three, of course, that we remember, main organs down here that secrete enzymes for digestion. And well, we have the spleen, which also secretes some hormones. Then we have the stomach, which holds within hydrochloric acid as well as pepsin, which help to digest, and the intestines, small and large, and the cecum in rats. There is also a cecum in humans. It's very, very diminutive. And in humans, we have the appendix. Use of the appendix in humans is currently being debated. Last time I heard it was not really used at all. It was just something like to help our ancestors chew grass or something. I'm not really sure. But the cecum, as we probably saw before, holds the bacteria which help digest food in animals. Last but not least of the major cavities, we have the pelvic cavity. And as you know, the pelvis is right here, this portion of you, and it holds your pelvic organs and stuff. These include the descending colon, the rectum, which holds the feces ready to be excreted, the bladder, which holds urine ready to be urinated, and the anus, which is the excretory organ, the ovaries in males, and the females, the prostate in males. And this is true for both male humans and female humans and male rats and female rats, which is what I'm trying to say. That it's the same for both of those. Same exact structure. More in common between us than you might have thought. So you might ask me, Felix, how about the brain? What's that in? The answer is a cranial cavity. And of course, humans and rats both have crania and cranial cavities. So there you go. That's one and the same. The spinal cavity holds the spinal cord if you want to stretch that far. And that's about it for cavities. One big difference between humans and rats in terms of their systems is that humans have sphincters, which are, of course, the little things that close off the stomach on both sides. It's like to let food come in and not. Rats don't have these. So it is always important to distinguish between chemical and mechanical digestion in our digestive systems. There's really only one really principal part of our digestive system that does mechanical digestion. That is the teeth. They grind the food up so it's really small. Then it can be brought down into our esophagus, into our stomach, through our intestines, and out through our ani. Now, you might think, well, Felix, doesn't the smushing of the churning of the stomach, isn't that technically mechanical? Not really, because the enzymes in the stomach, the pepsin, is the stuff that is digesting the food, and that's making it smaller. And this churning is just helping to spread it all out. And in the intestine, even though there is the paralysis and the squeezing and stuff, that's not really mechanical digestion. So there you go. Of course, one of the main underlying concepts of biology is how form can fit function in any given organ system. Organ, tissue, regardless. Well, at the moment we're talking about the digestive system, so we're going to talk about how the form fits the function. First, we have the fact that it starts out with mechanical at the top, then becomes chemical towards the end of the digestive system. This is very practical because you get the heavy lifting done, so the small, precise work can be done down at the bottom. Because if you didn't have any mechanic stuff up here, you wouldn't be able to. It would take so long for all those enzymes to digest all those huge macromolecules and those, all that stuff together. Plus, the surface area wouldn't be as big. It'd have to be more emulsification. It would just be hard over in general, Secondly, in animals like the koala bear with the especially large cecum, this is a fitting because they need to digest all the cellulose in the plants. And obviously, they need a very long tract in order to get all this digestion done. Similarly with the multi-stomached cows and things like that. And another very important part, of course, is the tiny little villi things on the tip of the intestinal bumps, which are very important because these make the surface area so much larger. There can be so much more digestion done within there. And that is the final part of this week's of studying. Thank you for watching. We'll see you next time, Mr. Hermanson gives us an assignment.